Okay, so we're going to wrap up the Euglenids here. I did want to show you some better pictures in this before I jump into Kinetoplastids. So here's just a drawing diagram, nice picture from the book, showing the general features and structures. Like I said, these guys all have two flagella. There's the main one. There's a tiny second one that is basically hidden almost completely within the anterior end of the organism. So it's why it's not visible on the exterior side. But they're all going to have mitochondria because that's part of endosymbiosis for these guys, the eukaryotes, etc. That endosymbiosis, they picked up the mitochondria, but then they also have the chloroplast as well. So a couple big key features. Oh, and the other thing to mention, make sure actually I'll highlight it, is the stigma. So that little light sensing structure right there is how they detect light levels. Again, they're not seeing images, it's a light level detection. So, okay, so as I mentioned earlier, some can be autotrophic, some can be heterotrophic, some are both. They run both methods. Light's available, let's move to the light and let's run photosynthesis. If we can't move to the light, let's just eat stuff. So again, big diversity within this group. I'm going to see if I can pull up this little video. Shows you guys how much diversity there is within the protists.
Euglena does not have a rigid cell wall, usually referred to as a pellicle.
Okay, so I'll narrate. That's hard to hear that. So the way this is moving is because they don't have a rigid cell wall, they have that pellicule. So it allows for flexibility and kind of this undulating movement of the organism, and then they're using those flagella to propel themselves. This particular group, both flagella are very, very obvious. Others, that second flagella, again, is kind of hidden and buried within the organism. So, okay, so just a little video there to give you guys something visual to check out when we're looking at euglenids. Again, grab a water sample if you have aquatic body of water. That's a silly, stupid statement there. If you have a body of water near you, grab a scoop of it, especially in the early part of spring when the weather's warming up and things are starting to get going, and check it out. It's crazy how many of these will just be swimming around in Lake Springfield, Taylorville's lake, Petersburg's lake, Lake Michigan, etc. So check it out. Like I said, they're good things to have in your water. Don't freak out if you're swimming and you know there's euglenids in the water. They're good. They tell us water quality is good. If they disappear, that's when you should stop swimming. When they're not there, because then there's a problem with your water quality. So, okay, the second grouping underneath the euglenids, or euglenozoa. Euglenozoa is the group, euglenids are the subgroups, and I'll diagram this in a minute here for everybody. The second one are the kinetoplastids. Okay, so these are nasties. These are things we don't want. Big feature of kinetoplastids is that they will have a single mitochondria. So within the organism, there's going to be a mitochondria that is the powerhouse. No photosynthesis, completely heterotropic here. These guys cause a variety of diseases. The trypanosomes cause what's known as African sleeping sickness. This is transmitted by the tsetse fly. That's a little fly down here. The fly is simply the carrier or the vector for the disease. The disease is caused by a trypanosome, which is a type of kinetoplastid. Leishmaniasis disease, Chagas disease, these are all protist-based diseases versus the viral, fungal, bacterial, etc. diseases we've discussed and will continue to talk about throughout the semester. So again, bio majors who are heading towards healthcare, you have to know the cause of the disease to treat it correctly. If you misidentify what is the cause of the disease here, you're not going to have an effective treatment. And sometimes you give the wrong treatment to a disease, you actually increase the problem. You give the wrong antibiotic to bacteria, they eat it and they make more bacteria. You give the wrong medicine to one of these diseases, it can cause damage within the human body and you're not getting rid of the disease. So very, very important for those of you heading towards healthcare. You have to know these problems and you look at symptoms. Challenge is lots of the symptoms overlap. So I'm not going to ask you guys to know all the features of the diseases. That's not the scope and the nature of this course. But let's look at some application to this. And again, think about how you would, in the future, work with this. These diseases are not restricted to just other countries. They are carried to the US, or when we travel, we have the potential for exposure. So we can't ignore diseases because we think, oh, we'll never be exposed to that. Climate change is causing all sorts of diseases to spread way outside their historical ranges. So, okay, so we're going to talk about African sleeping sickness, the trypanosomes. So trypanosomes are crazy little protists that can evade the host immune system by changing their cellular proteins. So the immune system responds by looking at a cell, let me draw it here, and we have these little protein markers, whether it's a virus, a bacteria, or whatever. There's the thing invading, and here's the immune system. White blood cells. They're out there defending your body. And they're looking at these markers, and they're saying, huh, you don't belong here. It's like checking an ID. You don't belong here. I'm going to attack you and kill you. That's the immune response, trying to destroy the invader. Well, once you've attacked and killed an invader once or twice or 3 times, your immune system gets smart and it knows how to tackle this and kill them the next time they show up. Well, trypanosomes switch out their proteins. Once the immune system starts learning, trypanosomes go, yeah, well, let's try this. We're going to go and instead of those blue ones, we're going to use green. And we're going to change the shape of it. And now we still can get in and attack, but you can't find us. You can't defend against us. So your immune system goes, huh, now I have to relearn what these are and figure out how to destroy and attack this. So it's constantly a problem for the immune system. It's always playing catch up. It can't get ahead. It can't get ahead and destroy the pathogen because a pathogen keeps changing its defensive strategy. One trypanosome can produce millions of copies of a single protein, so it allows it to have so much diversity when it comes to protein structures. It's just unbelievable. That's why it's a constant shift and change and adaptation by the parasite, by this trypanosome. Uh-oh. So these frequent changes prevents the host from developing immunity to the particular pathogen. So it's a huge problem. We see millions of people infected with this yearly. We see, unfortunately, thousands and thousands of people dying from it. They're basically destroying your red blood cells and the blood cells in the body. Let me go back here briefly. This is the trypanosome. These are your red blood cells. So they'll be swimming around in your bloodstream and they will attack and destroy your red blood cells. So you destroy all your red blood cells, you have a world of hurt. So that's the problem with trypanosome. So again, I encourage you guys, start thinking outside the box. What could I do? What could the medical community do to prevent this? To save lives? Do you focus on the vector of the disease, the tsetse fly? Do you focus on blocking this from entering the host cell, the red blood cell? Do you figure out how to bolster the immune system to destroy these things? There's 101 different things we can do. We need to do 110 different things to try to stay ahead of it. Because once we hit it with 101, it's going to evolve and adapt and mutate and change to go around our defenses and our strategies. So we constantly have to be working with this and trying to make sure we can provide as much help as possible. So, okay, so here's a review slide. I would encourage you guys, write this out. Don't print it. Write it out. Take 2 minutes, write out supergroup excavata, and indicate the key feature. Now, once you're in excavata, you go one of 3 routes, but you have to have that feature to go to the next route. So here's step 1, step 2, based on your feature, you go diplomads, you go to parabasolids, you go to euglenozoa. What are the features under each of these? Once you're in euglenozoa, you have 2 options. You go to Euglenids, Kinetoplastids. What are the key features for Euglenids, Kinetoplastids? So can you fill this out without using your notes? This is a great, notice a big hint here, this is a great review for the quiz, for the exam. Can you do this without your notes? If you can't, go look it up. But this is a great practice slide as you guys get ready for the Protiste exam. So the Protiste exam is challenging because it's brand new information for nearly everybody. And there's a lot of stuff in the Protiste world here. So this is just the first of the supergroups, excavata. We're going to now move our way into Chromio Alveolids. That's going to be our second supergroup. So I want you to be comfortable with excavata and all the features and characteristics there. Now let's move over to the supergroup Chromio Alveolids. So we are right here. So we did excavata, check. We're now going to tackle Chromio Alveolids. Okay, so to be a Chromio Alveolid, big key feature, and actually, yeah, drop the hashtag, we'll get the star in there. The big key feature is that these organisms may have arisen by one or more secondary endosymbiosis events. That is what places you in Chromio Alveolids. That is a key feature there. Within Chromio Alveolids, we have 2 key branches or subgroups. The alveolates and the Straminophila. Those are the 2 sub-branches or subgroups of Chromio Alveolids. So if I had an organism in front of you and you saw a groove in the side of it, you'd hopefully say, oh, excavata. If I had an organism in front of you, you're looking at it and you're like, okay, one or more secondary endosymbiotic events because it has these characteristics and features. That should put you in a Chromio Alveolids. Once you're here, we're going to branch down into, again, these 2 subgroups, alveolates, Straminophila. We'll start talking about these in the next lecture.